The effectiveness of a vaccine can be enhanced by targeting it to sites of antigen presentation. An important way of enhancing the effectiveness of a vaccine is to target it efficiently to antigen-presenting cells. This is an important mechanism of action of vaccine adjuvants. There are three complementary approaches. The first is to prevent proteolysis of the antigen on its way to antigen-presenting cells. Preserving antigen structure is an important reason why so many vaccines are given by injection rather than by the oral route, which exposes the vaccine to digestion in the gut. The second and third approaches are to target the vaccine selectively, once in the body, to antigen-presenting cells and to devise methods of engineering the selective uptake of the vaccine into antigen processing pathways within the cell. Techniques to enhance the uptake of antigens by antigen-presenting cells include coating the antigen with mannose to enhance uptake by mannose receptors on antigen-presenting cells and presenting the antigen as an immune complex to take advantage of antibody and complement binding by Fc and complement receptors. The effects of DNA vaccination have been enhanced experimentally by injecting DNA encoding antigen coupled to CTLA4, which enables the selective binding of the expressed protein to antigen-presenting cells carrying B7, the receptor for CTLA4. A more complicated set of strategies involves targeting vaccine antigens selectively into antigen-presenting cell pathways within the cell. For example, human papillomavirus E7 antigen has been coupled to the signal peptide that targets a lysosomal associated membrane protein to lysosomes and endosomes. This directs the E7 antigen directly to the intracellular compartments in which antigens are cleaved to peptides before binding to MHC class II molecules. A vaccinia virus incorporating the chimeric antigen induced a greater response in mice to E7 antigen than did vaccinia incorporating wild-type E7 antigen alone. A second approach is the use of ISCOMs, which seem to encourage the entry of peptides into the cytoplasm, thus enhancing the loading of peptides onto MHC class I molecules. And that's the effectiveness of a vaccine can be enhanced by targeting it to sites of antigen presentation.